Right. Shall we get started? Night. I'm excited to get bringing Common Science Bubble to North Branch. North Branch, thanks for hosting us tonight. So I'm going to go ahead and read our land acknowledgement and then tell you a little bit about Common Science Bubble series. Berkeley Public Library buildings are built on the territory of the shown within the homeland of the Chochenyo-speaking Ohlone people. We acknowledge and honor the Ohlone people who have lived in Berkeley and throughout the East Bay since time immemorial. The library recognizes that Berkeley residents have and continue to benefit from the use and occupation of unseceded stolen land. And we pledge to recognize the permanence and the rich contributions that the Ohlone people continue to bring to the city of Berkeley and throughout the East Bay. We celebrate the strength of the Ohlone people and their culture. Berkeley Public Library is committed to helping create a more just and anti-racist society and to furthering the visibility and celebration of Ohlone authors who are specific to the East Bay and Ohlone culture in our collections and program. This is a living document that was co-written by Ohlone cultural leaders Vincent Medina and Louis Trevino and the Berkeley Public Library. And if you're interested in learning more about Ohlone culture, we have a monthly Cafe Ohlone series. It's really cool. Happens on campus near the First Anthropology Center at Cafe Ohlone. Vincent and Louis talk about living Ohlone culture, and they usually serve Ohlone stems and talk about the ingredients. So it's pretty neat. And you can find that on the library calendar. It's usually the third Wednesday of the month at 3:30. So welcome to Talking the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or topic that they find really interesting. The organizers are 2 graduate students at UC Berkeley, Kaylin Schwang and Daniel Brethow. This program happens every 3rd Tuesday of the month at 5:30 at the library. Right now, Science Bubble is on tour, so we're going to be at the different branches. Next month, we'll be at Claremont Public Library, and it's always on Zoom. If you're interested in checking out their past seminars, you can visit their website and sign up for the listserv to keep in the loop for the upcoming seminars. Also, check out their YouTube page. The Science Bubble YouTube page has a fabulous archive of past programs. They're really interesting, and you can see this lecture as well. All right, thank you.
Thank you so much, Kelsey, for the introduction, and thank you all for attending today's seminar. We're very excited to be here today. This is, as Kelsey mentioned, our stop 1 of our library tour at the North Branch. So to follow up on what Kelsey had mentioned, at the end of our talk today, we'll have a QR code to scan and join our listserv. You can also go to bit.ly/TSE for Talking the Science Bubble to join our listserv, and you can visit our website or go directly to our YouTube account if you would like to check out the previous videos. And so for tonight, we're very excited to have Matangi and Tiffany to be our speakers. And this is an open format talk, so please feel free to raise your hands. And for folks on Zoom, feel free to type in the chat or use the Q&A to submit questions, and we will ask them in real time. And so without further ado, it's my pleasure and honor to introduce our first speaker for tonight, Matangi Kumar. Matangi grew up in Syracuse, California and has always loved biology. She went to undergrad at UC Davis, just nearby, where she majored in genetics and genomics. She then started her PhD at UC Berkeley in the Vision Science program in 2021. Matangi is currently a fifth year in the program, anticipating to graduate in the spring. Exciting. In her free time, she loves to hang out with her friends, and she's also part of the Taekwondo club on campus and loves arts and crafts.
As Caitlin said, I'm Matangi. I'm a fifth year at East Berkeley, which is wild to say because it feels like I just got to the campus like not so long ago. But I'm part of the Vision Science department here at Berkeley, and the work that my lab does is study glaucoma and neuroinflammation within the retina. I'll be describing all those things in my talk later on. But yeah, my talk is the eye under pressure, glaucoma, and neuroinflammation. So what is glaucoma? I was told that this is an interactive presentation, so if anyone knows what glaucoma is, take the board. But glaucoma is a group of optic neuropathies leading to the death of retinal ganglion cells. So some of the risk factors that we usually see are age. So glaucoma tends to affect the older population, high ocular pressure. So a lot of us think that glaucoma is caused by ocular pressure in the eye, but that's actually just one of the main symptoms that occurs that could lead to glaucoma. Race and genetics also play a big role in glaucoma. There are certain ethnicities that might be more prevalent in getting glaucoma, but what we categorize as glaucoma is the death of retinal ganglion cells. So those are these cells that we see right at the bottom of the retina here. And it's actually one of the leading causes of irreversible blindness worldwide. So going into more depth on the retina, so our retina is this layer of tissue that's light sensitive near the back of our eye, and its main job is to convert light into electrical signals that go to our brain to help us see. So to simplify the retina right now, we have three main types of neurons that transmit electrical signals to each other that connect to the brain to help us see. So light goes through the lens right here, through the retinal ganglion cells, bipolar cells, and photoreceptors. And our photoreceptors take this visual signal and send it all the way down to the bipolar cells and retinal ganglion cells to the brain, and that helps us see. But what happens in glaucoma is the retinal ganglion cells are the main ones to die. So as the electrical signal go through, it stops that transmission to the brain, and that's why we lose our vision. And how do the retinal ganglion cells die? So in this context, I'll be talking about cases of glaucoma that have elevated ocular pressure. So this pressure on the eye, it tends to strain the optic nerve or the optic nerve head at the most. And all of our axons, so those are the end portions of our neurons, they lead into the optic nerve and help us see. So this optic nerve head is the most vulnerable area for our retinal ganglion cells, and that's where all of our axons lead out to. So if this is a retinal whole mount, so if we've taken a part of the retina to lie flat so we can observe how our retinal ganglion cells die, it's at the periphery with ocular hypertension that these retinal ganglion cells start to thin out, and that slowly causes our vision to deteriorate. So our retinal ganglion cells are, one, the most vulnerable because their axons exit through the optic nerve, and two, the peripheral retinal ganglion cells are the first to die because their axons are so much longer, and so it leads them to not get enough nutrients to support them. So because our peripheral retinal ganglion cells are the first to die, that's actually how we lose our vision too. So we start losing our peripheral vision first. So if this is our clear 20/20 vision as glaucoma progresses, we start to lose our peripheral vision first as it moves just to our central vision. And this is actually one of the main reasons why glaucoma is very hard to diagnose at early stages. because we have two eyes and they're working together to help us see, we kind of like don't tend to recognize when we're starting to lose like just our mild peripheral vision. It's not until we lost a decent amount of our retinal ganglion cells that we start having any like actual visual defects that prevent us from performing day-to-day actions. I do have a video, but it's not working, so I was told I would be sending that out. So it's like a simulator to show how someone with glaucoma kind of goes about the world. Right now, the treatments for glaucoma are actually just to lower this high ocular pressure. There are a lot of different drug classes. I just put this slide on here so people can have access to see what are the available medications and drugs right now on the market, but I won't delve too much into detail. But because the current treatments are just to lower this ocular pressure, this actually just slows the death of our retinal ganglion cells, but it doesn't prevent them from dying. So as the disease continues to progress, you still see people's visual field start to deteriorate. And so the search right now is to find a neuroprotective treatment so that it preserves the retinal ganglion cells the moment you're diagnosed and you preserve that vision at that moment of diagnosis. So that's the work that my lab focuses on. So I'll move from more background into just some basic neuroprotective strategies that my lab is working on. So we identified a lipid mediator compound called lipoxin B4, and we found that it's neuroprotective. So we have specific types of cells in the retina called astrocytes, and we found that these astrocytes naturally, as part of its function, produce these lipoxins, and we found that that is protective towards retinal ganglion cells. So we tested this in a mouse model, and so we're looking at the thickness of our ganglion cell layer over here, and when we have ocular pressure, it thins like very significantly. And so this is where we start losing our visual function. But when we treated our mice with these lipoxins, we see that we preserve this ganglion cell layer. So it's shown that this is an adequate strategy in order to protect our retinal ganglion cells from dying. And another thing we see is in the green, these are other support cells like astrocytes that I mentioned up here. And when we have ocular pressure or any kind of stress in the eye, they light up super bright green, and this can lead to some like destructive pathology within the retina. But when we treat it with lipoxins, we've kind of preserved the reactivity of our astrocytes. And so I wanted to talk about different ways that we can study glaucoma in a lab. So it's difficult to study it with humans, and so we use mice as a way to model ocular hypertension. So what we do is we have mice, and then we induce, so we put silicone oil in the lens, in the cornea of the eye, and that kind of creates this super big pressure environment, and this is how we inject the mouse with silicone oil, but we see like a droplet that occurs right in its cornea, and that's how we know that it has ocular pressure. So the way you go to the same way that you go to an eye doctor and you get your eye pressure checked with that like pressure machine that touches the tip of your eye with a puff. We do the same thing with the mice. So that's called a tonometer, and we measure the ocular pressure with the mouse too. And so we show here in the black, like this is a control mouse, so a mouse that we haven't induced ocular pressure into, and the red, this is where we've injected silicone oil. So you can see significantly how our mice that have the silicone oil has significantly more ocular pressure. And this is the way we study it in mice. And one thing that our lab specifically studies is we don't really study neuronal function. We study something called glial cells in the retina. So these are our retinal immune cells, and they're our retina's defense system to fight against inflammation. And so we have three kinds of glial cells in the retina. So we have astrocytes, malar glia, and microglia, and they all have distinct roles in order to help our surrounding neurons in order to help them see. And when there's stress in the eye, like ocular pressure, they become reactive. And so they're responding to the stress the same way, like if you were to get a cut, your immune cells respond to that kind of chronic inflammation that's occurred. And so when our cells become reactive, they can have destructive effects on our retina as part of their immune signaling. And for my project, I've been studying Mueller glia and its roles in diseases like glaucoma. And the way that I can study Mueller glia and glaucoma is I can look to see if they can produce lipoxins. And the way I can study that is I actually extract the Mueller glia from the retina, and then we can run all kinds of assays on the cell in order to see like how it kind of reacts to stress, like ocular pressure, how it reacts to different acute stressors, and how its lipid levels change like between these conditions. And so this is kind of just like what my thesis project has been, but I studied Mueller glia and I found that they make lipoxins, which is a homeostatic function to protect against glaucoma. My goal in this project is to see how they function when they become reactive, and then to see if we treat our mice with lipoxins, how this kind of affects Mueller glia. Like, does this bring it back to a state of resting? And does it create like homeostatic functions again for the rest of the eye? These are some of the different scientists that I work with in my lab. So this is my PI, Karsten and John up here, and two of the postdocs in my lab, Shabir and Shivam, and they've basically been my mentors for my entire PhD project, and kind of helped me get to the work that I've done today. Yeah, that's my talk. If people have any questions.
I saw in your funding source, everything was NIH.
It's not the best right now. Luckily we had some alternative grant sources from the, so that's kind of been operating, but we're kind of just crossing our fingers to hope that the next grant cycle that we submitted our grants to might get funded, but it's been a tough time to secure funding for these projects.
So if I understand, it's my impression that your ocular pressure can change throughout the day. So is it a case that when it's super high, I think in the morning, that your glaucoma symptom would be worse? Or is it like sensitive enough to the day-to-day exposures?
So let's say you're going to like the optometrist for your regular screening. Depending on the time of day that you go into, if pressure is abnormally high in the morning, they might ask you to reschedule your visit for later in the day just to make sure that that's not an effect that you're seeing, but you're chronically having pressure throughout the day. And so we have fluctuations, like even just like our sleeping position can affect eye pressure, but if our eye is functioning properly, that shouldn't be affecting the state of your retina. It's when you have this chronic pressure that's not going away that causes kind of the symptoms you see.
So chronic pressure usually like is sole cause of like the retinal ganglion cell death, or is it usually kind of like prolonged something else?
Yeah, so for open angle glaucoma, so that's the most common type of glaucoma that people see in the clinic. Usually ocular pressure is like the first symptom they see, and that causes like different vascular and metabolic like changes. I think because glaucoma is a disease that affects like the older population, there are just a lot of comorbidities, like different vascular diseases that could play a role in it, but there are also different types of glaucoma. So there's different, like there are types of glaucoma where you don't even have pressure in the eye, but we still see the retinal ganglion cells die. And that's probably just from like different stressors or like vascular insults that happen that's causing just the retinal ganglion cells to die. But it's kind of like a why are they dying from pressure? Even though when we stop the pressure, they still keep dying. That's kind of a mystery question.
Great talk. I'm not too familiar with glaucomas, but is this more like it's kind of like predisposition or more like an environment?
So there are genetic factors. I think there are a couple of target genes that they see in like cases of juvenile glaucoma, so they call it when you get glaucoma before the age of 40. But it's more environmental. Like if you have like high myopia or meaning if you're super nearsighted, like that could be a risk factor. While there are like some genetic hits, like it could just make you more likely to be predisposed. It's not like a one-to-one correlation. Like you have the gene, you will get. It's like a combination. So
One quick clarification, so we were adding the toxin, I think it was called. Was that after glaucoma had begun to set in, and so it's still protected, or it was like after some of the cells have been damaged, and then it kind of like helped to re-thicken the optical?
yeah, so from our like published work, so what they've done is after like 4 weeks of inducing ocular pressure, they started the treatment. And you can't replenish the cells that have already died, but you're kind of preserving it so that it doesn't die.
I don't know if you know the answer to this, but when you're, I guess, giving the like toxin back into these like glaucoma like retinas, I assume like if you were trying to do this in people, you can't really do that. Is like, have people looked at how one would like introduce like toxin as like a treatment?
Yeah, so when we do it in the lab, we give eye drops and we give like an intravenous injection also, but the goal with this research would be to create some type of gene therapy that allows us to like make the eye like make more like toxins through like a viral vector. So that would be the goal. That's what we're working.
If intraocular pressure causes glaucoma, is that a physical attack on the retinal ganglion cells? And then if that's true, does lipoxin give it a suit of armor, or is it something internal to the retinal ganglion?
That's a really good question. So we have IOP lowering drops. So that would be fighting against the actual ocular pressure that's occurring, and that's available on the market. But when you're getting a lipoxin treatment, I'm actually not sure whether it's like helping the ganglion cells like, remake its own like lipoxin circuit so that it's preserving itself from the insults, or whether it's like a shield. So I think we would have to look at like the mechanism of how the lipoxins are interacting with the rest of the ganglion cells.
So if it's not pressure that's causing it, the lipoxin may or may not help with glaucoma switch.
So I would think of it as like, even if pressure wasn't the case, the cells are still dying. And so what the lipoxins are doing is just prevent that from continuing to die.
Full disclosure, glaucoma.
Oh,
Normal tension glaucoma.
oh, so you don't have ocular.
Who knows?
Do you have any questions on this?
We do not, actually. Yeah, but we do have 9 attendees, actually, so. Yeah.
Let's give another round.
And I will introduce our second speaker while we're switching over laptops. So our second speaker is Tiffany Tran. And Tiffany grew up in the suburbs of Los Angeles, California, and attended the University of California, Riverside. where she started pursuing research in a materials and organic chemistry lab. She earned a bachelor's of science in biochemistry in 2020 and decided to pursue a neuroscience PhD after becoming intrigued by the idea of non-invasive control of cellular activity as a therapeutic intervention. She decided to join the neuroscience graduate program at UC Berkeley to investigate how changes in cellular activity in the brain could affect our cognitive behavioral functions using relatively non-invasive techniques. Outside of research, Tiffany enjoys cooking, hiking, listening to music, and exploring new cafes and restaurants. Thank you so much for being here to speak with me. Take it.
Thank you all for being here and thank you for the kind introduction. My name is Tiffany. I am a neuroscience PhD student at UC Berkeley. I'm also affiliated with the Department of Electrical Engineering and Computer Sciences, part of the LEU Lab. And in the LEU Lab, we do a bunch of magnetogenetics sort of experiments. And today I'd like to take this opportunity to talk to you all about these set of techniques in which we can modify cellular activity using magnetic fields. So since I'm a neuroscientist, I'd like to probe for a couple of volunteers or so, as I ask you all to think about what do we know about the brain.
I think that's where I am.
I think therefore I am. That's a great one. Other ones? We have neurons. We have neurons. Great. We're going to talk about neurons shortly as well. But also that the brain is responsible for a bunch of our functions, right? So different parts of our brain are responsible for processing our senses, such as smell, taste, touch, as well as our sights. There are regions, specific regions in the brain that are responsible for emotion, as well as our thoughts, decision makings, and our memories. And so the brain is responsible for pretty much everything, how we live, how we perceive the world, how we react to the world, and how we remember the world. And so all of this is very high level. At the core of it, all of this is done through the activity of cells. And one type of cell is the neuron. And so neurons are the cells that kind of receive and process and then send the information, these signals that they obtain from the environment to nearby cells or to each other. And so these are very, very small cells. They're 300 to 400 microns long, usually. And so if you think about thickness of your pinky, that's about maybe a centimeter. And so these neurons are up to 10,000 times smaller than what your pinky's width is. So they're very, very small. And you would definitely need a microscope in order to view them. However, the morphology behind these cells kind of plays into all of these roles that they can do. And so here I'm showing a really cute cartoon by Christina of dendrites, which you can think of sort of like the branches of a tree. They're very spread out and they can interact with the environment and collect signals from the outside part of the cell and integrate that. And if the cell determines them to be important signals, they'll send that down the axon, that part of the cell, and then send those that information to the nearby cell. It allows us to interact with our global environment, things that we interact with in the world. And so, like Matangi had mentioned, there are also other cells that kind of support neuronal functions, and they're called glia cells. In the brain, there's quite a variety of them, including microglia, which are resident immune cells. You think of them sort of like little doctors or little like vacuum cleaners. They identify what's foreign and shouldn't be there, and essentially gets rid of them to maintain the integrity of our brain. We also have oligodendrocytes, which essentially allow for faster communication between neurons. And there's obviously blood vessels that are going to be allowing for oxygen and nutrients to get into our brain. Today, I'm going to focus mostly on this other type of glia cell called astrocytes, which Matangi had also mentioned very briefly previously. And so what are astrocytes? Astrocytes comes from the Greek word astron, which means star, as well as cytos, which means cell. And so these cells are star-shaped. They are known to play a variety of functions. You can think of them as like the jack of all trades of the cells. And they can regulate neuron-to-neuron communications, they can supply nutrients, they modulate the blood-brain barrier. And so there's so many functions that these astrocytes can perform, but we don't exactly know how they perform them. There is this hypothesis that changes in calcium levels inside of the cell actually contribute to all of these different functions that the astrocytes perform in the brain. And so in order to investigate how astrocytes actually function and could support or even play a more active role in the brain, we would need to be able to monitor the calcium activity. And so one way we can do this is through this experiment called calcium imaging. And in our lab, essentially, we will have a set of cells that we place under the microscope, which is hooked up to a computer that we can record their activity. Astrocytes would then express this calcium indicator. So essentially, when there's an increase in calcium levels, this would bind to this indicator called GCAM, and there would be this green fluorescence, this green shine. And what we would see under the microscope is something like this. And you can see how in certain areas of the cell, there's going to be this increase in calcium levels, which corresponds to this increase in the green fluorescence inside of that we're watching here. And not only is there an increase in one part of the cell, but that increase can spread to another part of the cell and spread to the other cells as well. So this is a very complex and dynamic kind of phenomena. I think it's pretty cool. I think it's probably one of my most favorite parts of doing these type of experiments. It's watching this phenomenon, which is also referred to as calcium waves, which is aptly known, or aptly named rather. And so now that we have this set of techniques that we could use to monitor the calcium activity, if we wanted to alter their activity in order to investigate how astrocytes actually, function in a circuit, we would need to be able to kind of probe it somehow. And so one way that our lab uses is through magnetic fields. And so by applying an electric current into a coil, you can think of this sort of like as a copper ring. If we apply an electric current, we are able to generate a magnetic field through the coil, and that could be applied to the to the cells that are magnetically susceptible. So they would probably the culture of cells would be placed above here, and you can see that the magnetic field would then interact with those cells. And so this is very useful because magnetic fields can be non-invasive. They can interact with biological tissue very effectively, and we can apply the stimulation on demand. And so if we wanted to remove this stimulation, all we have to do is remove the electrical current, and there would be no more magnetic field, and this allows for better control of the stimulation. And so in order to, get cells to respond to magnetic fields, we'll need to have them express certain channels that will be sensitive to the stimulation. And so this is sort of similar to how in the old days, we would have a TV box with a physical antenna sticking out of it. And so when we apply, or we turn on the remote, it would send a certain signal, radio frequency signal, and that would allow for the TV to turn on. In a similar manner, we're going to be using the same principle in what we call neurogenetics. In the neurogenetics, instead of radio frequency fields, we're using magnetic fields to modify cellular activity. The cells will need to express new channels, so the astrocytes will be expressing a new channel, similar to the TV needing to have those antennas present in order to respond to the magnetic field stimulation. So the TV remote, once we turn that on, when we apply the magnetic field stimulation, there would be this increase in calcium activity inside of the cell, and we would see an increase in green fluorescence. And similarly, or analogously, this would correspond to the TV turning on. And so what that would look like? Well, here I'm showing you a video of what this increase in calcium activity inside a single astrocyte kind of looks like. You can see that there's more frequent events, there's more flashes of green, and the events are more intense. So you can see that the green flashes are a lot brighter with stimulation versus without stimulation. I'll play that one more time. I've also quantified it here through this heat map. On the y-axis are the different events over time on the x-axis, and you can see with the warmer colors, there's a lot more warm colors on the RF, or the radio frequency magnetic field stimulation, compared to normal without stimulation of the conditions. And so, given that, what can we do from here? Well, essentially, I've showed you that magnetic field stimulation, magnetogenetics, could be used as a new tool to probe cellular activity and to modify cellular activity. So from that, we'd be able to study different cellular functions without actually disturbing their system. And so we can probe all these biological processes that could happen in the deep brain region and the cellular mechanisms that underlie that. And through all of that, in especially disease states, all of the information that we would gather from that situation, we would be able to use in order to treat And so we could develop drugs, medications, pharmacological interventions, as well as other therapeutic approaches such as directly stimulating the cellular targets using magnetic fields or other approaches. So all of these applications are something that I'm really interested in and what I'm looking forward to continuing to investigate for the rest of my thesis. Here are my acknowledgements. So this is the Liu lab. I am and Professor Liu is in the back there with his little fedora. He's my direct supervisor. He's been incredibly supportive. I would also like to thank my Magnetogenetics team, a group of very diverse scientists that have been pushing me, and I feel very inspired and motivated every day, just learning alongside them. I'd like to also thank the Pumping for Science Bubble Organizing Committee, Keelan and Daniel, as well as Kelsey in the Berkeley Public Library System for putting together this event, and for you all for taking the time out of your day to listen to our talks. These are my funding sources, and if there are any questions, feel free. Thank you.
You mentioned that like we use for deep brain stimulation, but what's that look like?
Yeah, so the idea is that we would have to still have those channels expressed in those regions, but once we do that through genetic approaches, what we would do is essentially have a foil that would have magnetic fields to be applied to, and then we would be able to turn on with the flipped switch, stimulate those regions, and observe how specifically those regions would contribute to certain behaviors or other great questions.
I'm curious about the genetic channel. So was that something that people found in like the mechanism, or did someone engineer it to be responsive to the magnetic field?
Yeah, so it was an engineered, so someone who had graduated from a lab had engineered and developed this ferric channel. And essentially there's a couple of papers that have been published already using this channel. And we're able to target it to specific channels to allow for specific ions to flow into our cell.
So does the like strength of the magnetic field change the frequency of the pulses? Like, can you adjust that, or is it just kind of like with the magnet it does this without the magnet it makes this?
That's a great question. So the strength of the magnetic field is different from its frequency. And so right now we're using a specific frequency, but other labs, other groups have also used different frequencies to stimulate their cells, although it's less than a different type of tool. In terms of like the strength of the magnetic field, we have shown that, or it has been found that increasing the strength of the magnetic field increases the efficacy of this tool. So there is some slight dependence on that. Whether there's a dependence on the type of frequency, I think it depends on the question that we're asking and what system we're using to control the cells.
How do you incorporate like the therapeutic approach with?
Yeah, so I think initially the idea was before the lab had created this was to be able to record and stimulate cells inside, like at the same time inside the MRI machine. So that was like the ultimate, like, this is gonna be super cool. I think that's still something that we're considering, but the idea is that if we are able to have a subpopulation of cells express this magnetic sensitive channel, we'd be able to modulate or like modify their activity in a way that could address, for example, in a disease state where there's like less activity, could increase their activity, and perhaps that would counteract any of the symptomology that it is.
It's been done in vivo.
We're in the process of doing it. So super exciting. Stay tuned.
Just like, I think there's different types of these like magnetic channels and the fine.
use them at the same time? Is it like, are there different sensitivities and then they operate like about the same?
That's a great question. I think right now we don't have the answer to that. I do know that there have been some labs that have worked on a different, not specific to this particular tool, but another tool where they're able to modulate a subset of like magnetic nanoparticles, very small particles that are sensitive to magnetic fields. They're able to tune those to specific frequencies, and if you apply multiple different magnetic frequencies at once, you could get like simultaneous control of channels. But I think it's like a hardware issue right now. Thank you.
We have a question from Zoom. What sort of spatial resolution are you obtaining with your magnetic field generating mechanism? How field cells can be stimulated at once?
Great question. So spatial resolution. It depends on so the magnetic field can penetrate through the whole biological system fairly well without any significant attenuation. So pretty well. I think in terms of like cellular resolution, it depends on what cells are going to be expressed in these channels. And so it gets back to the genetic problems, having this channel well expressed, or how we don't want to like overexpress the system either, because in that case, it's possible that we're not controlling some or like not modifying a certain subset of cells, we're just overloading the system. Yeah. So it really depends on the expression of these chunks.
Is this something that is usually used like in the lab, or are there like therapeutics for diseases that can use this? Because I don't know, like introducing a new kind of channel into cells, is that like, I don't know.
Yeah, no, that's a great question, a very good point. So currently, I think we are all of the magnetogenetic approaches have been mostly just lab bench wise. It hasn't been any that I know of, any clinical applications yet. But I think it's really possible. It just depends on whether or not we control again the expression of these channels and not overexpress them or cause other potential bad effects from having the system expressed.
What is the benefit of like magnetogenetics and compared to like other cell manipulation techniques like optical?
Yeah, so that's a great question. So in the case of, for example, optogenetics, which uses light in order to modify cellular activity, magnetogenetics uses magnetic fields. And so the main difference between light and magnetic fields is that light can scatter in biological tissue, especially if they're in deep brain regions. But magnetic fields, again, they're able to penetrate through our bodies pretty well. And so we're able to reach deep brain regions without needing to like perform surgeries and add an electrode into the brain.
Kind of following up on that question, is there a reason why you would use magnets instead of just like an electrical like pulse?
Great question. So I guess it depends on the system that we're looking at. Because we're looking at the brain and there are neurons which are electrically active. If we apply electrical pulses, they could potentially interfere with the electrical communication of neurons. I think that's like my first line of thought. Yeah, I think that's the first line of thought.
Questions? Then let's thank our speaker, Schultz.
Thank you so much to both our speakers for very interesting talks tonight, and I realized the theme kind of lined up with the glial cells and the astrocytes, so that was very cool. And yeah, so this is our QR code. If you're interested in today's talks, please and want to hear more and join our mailing list. We send approximately 2 emails per month, and so in the next week or so, we'll send out an after seminar email with the video that Matangi had mentioned, as well as some other resources that our speakers have shared with us previously. And our next seminar will be on Tuesday, October 21st at 5:30 p.m. and will be at the Claremont branch. And these are our 2 speakers and their topics, chemistry and astronomy. So very exciting, and we hope to see you then. Thank you so much for coming.
Thank you, everyone. Yes. And thank you, North Branch, for hosting this too.
Yes, thank you so much.
So do we need to put back the ring on that square configuration there? You don't need to.
Chairs back along the end.
I'll always help with that. Don't want to, whatever now. Yeah.
I was like, texting my friends, like, check these out. So. If somebody said there's a house or a.
Yes, yeah, the science bubble, science bubble. So great. Yeah. Yeah.
I was like, I am live tweeting this.
Yeah. It's. It's on all the time. Yeah. Yeah, it was great. It's like super fed. Yeah. And the speakers, like, it's really nice because.
Yeah, you send out for email.
Yeah.